Hello, my name is Katerina Jaworsky, and I am a third-year medical student here at the Kirk Kerkorian School of Medicine at UNLV. Today, I will be presenting my poster about the research that I conducted with my PI, Dr. Arpita Basu from the Department of Kinesiology at UNLV, titled Diet Quality During Pregnancy and Cardiometabolic Risks in Women with Obesity. The objective of this study was to identify associations of maternal dietary habits with cardiometabolic risks in minority women who are inherently at higher risk for metabolic complications of pregnancy, which include gestational diabetes, abbreviated GDM, and hypertensive disorders of pregnancy, abbreviated HDPs. These metabolic complications can have numerous consequences for both the mother and the child, including birth complications and increased risk for type 2 diabetes in the mom later in life, as well as shoulder dystocia and postnatal hypoglycemia in the fetus. Thus, looking at dietary and lifestyle modifications during pregnancy is beneficial to potentially decrease the burden of these metabolic diseases of pregnancy, as well as provide a more feasible option for management of these conditions. This study was a secondary cross-sectional analysis with data from a previously reported study of 52 women who were recruited from the UNLV OBGYN departments here in Las Vegas. All women were considered obese as classified via their BMI, and were at high risk of GDM and HDPs based on their medical and past pregnancy histories. They were asked to maintain a food log for 2 weeks, which we then analyzed for nutritional contents, and blood samples were taken to analyze for various biomarkers. Linear regression analyses were used with a p-value of less than 0.05 determining significance. The two tables on the poster show the results of our linear regression analysis, which is reported as a beta plus or minus standard error. The bolded results are those that showed a significant relationship based again on the p-value of less than 0.05. Table 1 shows the association of dietary nutrients with cardiometabolic risks in women, which we measured via body weight, BMI, HbA1c or glycosylated hemoglobin, blood pressures, triglycerides, and HDL cholesterol. Our analysis demonstrated that an increased total fat intake was positively associated with classic risk factors for metabolic complications, such as increased BMI and body weight. In other words, an increased total fat intake was associated with an increase in BMI and an increase in body weight. Saturated fat intake was positively associated with an increased HbA1c. Both fiber and dairy actually demonstrated protective effects on the cardiometabolic risk factors for our metabolic complications of pregnancy, with increased fiber intake showing a negative association with body weight, BMI, and HbA1c, and dairy showing a negative association with systolic blood pressure. Table 2 below shows the associations between dietary nutrients and maternal biomarkers of inflammation, which we measured as C-reactive protein, adiponectin, interleukin-6, and others. Increased total fat intake was positively associated with an increase in C-reactive protein levels. Fiber and dairy again demonstrated protective effects, with fiber intake being inversely associated with CRP levels and dairy intake being inversely associated with IL-6 levels. Vegetable intake also demonstrated a protective effect, with vegetable intake being inversely associated with CRP levels. In conclusion, this study has demonstrated that maternal dietary intake modulates important cardiometabolic risk factors during pregnancy, specifically in minority women who are inherently at higher risk for these complications. However, it is important to note that the results of these studies are correlational, so these dietary nutrients and food groups must be further studied in clinical trials in order to determine causality between the maternal dietary intake and metabolic complications of pregnancy. Overall, our study has shown that it may be beneficial to consider altering the diet of high-risk pregnant women through decreased fat and increased fiber, dairy, and vegetable intake. Finally, I would like to thank Dr. Basu for her help with the research and preparing of this presentation, as well as for her continued mentorship throughout my research experience with her, as well as the UNLV GPSA for putting on this research forum. I wish I could be there in person today. Attached to the bottom of the poster are my references, and correspondence can be directed either to myself or Dr. Basu, and our emails are on the poster. Thank you so much for listening to my poster presentation today.